Today we will be discussing on the Indole test. So this Indole test demonstrates the ability of certain bacteria to decompose the amino acid tryptophan to indole that accumulates in the medium. Though this indole production test is very important in the identification of enterobacteria, and it is also a part of the INVIT procedure. So this INVIT stands for I stands for indole, M for methyl red, and V for Vox per skirt, and C for citrate utilization test. Now this I is a lowercase to ease the pronunciation. So this indole test is designed to distinguish among members of the family Enterobacteriaceae. Now, coming to the principle, basically this indole test is an example of biochemical test that depends on the biochemical metabolic characteristic. Indole test identifies bacteria either indole positive or indole negative based on their ability to convert tryptophan into a compound known as indole using a chain of enzyme collectively known as tryptophanase. Tryptophanase reacts with water, which is a hydrolyzed reaction or a hydrolysis reaction, along with tryptophanase enzyme to break tryptophan down to get three different products, that is indole, pyruvate acid, and ammonia ion, which is a kind of deamination reaction because it serves ammonia group from carbon, so it gives out ammonium ion. So this is the principle of indole test. Coming to the reagent that is used in indole test, here we have a COVAX reagent. This COVAX reagent constitute the p-dimethyl aminobenzaldehyde 50.0 gram and hydrochloric acid 37%, 250.0 ml, and amyl alcohol 750.0 ml. However, it can be adjusted or supplemented as required to meet the performance criteria. Now, instead, the alternate use for the COVAX reagent is Ehrlich reagent. This Ehrlich reagent is more sensitive than the COVAX reagent, yet COVAX reagent is most commonly more commonly used in the indole test. Coming to the procedure, first of all, you have to take a sterilized tube containing 4 ml of tryptophan broth, then inoculate the tube aseptically by taking the broth from 18 to 24 hours cultured, and then you have to incubate the inoculated tubes at 37 degrees centigrade, which is the body temperature, for 24 to 48 hours. So why do you have to keep this 24 to 48 hours is because this is to give the test organism the time to convert tryptophan to indole if it has an enzyme tryptophanase. Now after 24 to 48 hours, you are going to be adding 0.5 ml of COVAX reagent to the broth cultured, and then you are going to observe for the presence or the absence of a cherry red ring. Now coming to the interpretation, in indole positive, there will be a formation of a pink to red colored cherry red ring in the reagent on the top of the medium within a seconds after the adding of the COVAX reagent. This is because there is a presence of tryptophanase enzyme to break down tryptophan to indole. Thus, it gives the formation of a cherry red ring on the top of the medium, so your test organism can be reported as a indole positive. Whereas in the form of negative, there will be no color changes since there is no indole present because there is no tryptophanase to break down tryptophan. So this media will end up into yellow. So this media ends up being like yellow in color only rather than forming a cherry red ring. So one example of indole positive bacteria species is E. coli, and one example of indole negative bacteria species is Salmonella. So this is all about the indole test. It is a convenient test and easy to carry out.